Thank you very much for inviting me. I'm going to talk a little bit about my research in breath analysis, and I hope by the end of this lecture you'll think there's more to breath than halitosis. So, just so that you don't think that it started with my group, people have known about breath for centuries. In fact, Hippocrates used the exhalation of water as evidence of life. And people have taken the impressions of odors from the body, such as sweat, urine, breath, to diagnose disease. You can walk into a room and you can tell a person is diabetic. You can tell a person has liver disease. This is just straight away from looking at a person. And of course, Lavoisier measured carbon dioxide in breath. And actually, breath analysis in these days is actually coming with modern analytical chemistry. And one of the earliest workers in the field was Linus Pauling. Now, he published one paper, and it was a significant paper. So, okay, I thought I'd start off and give you some physiology and talk about breathing, because breath is related to physiology. So I took as a standard is a normal, healthy individual, a male, with about 75 kilos and 1.7 meters squared with a BMI of 25. And when you're breathing normally, you're breathing under autonomic control. In other words, you're not aware of what you're doing. And a typical person, this sort of person, will breathe about 600 mils in a single breath and will breathe 10 to 12 times a minute. At that rate, you have an anatomic dead space of about 130 mils. This is the sort of the plumbing in your mouth and your trachea. And you have alveolar ventilation of about 4.7 liters. This means that this person will inhale about 360 liters in an hour. So you are essentially, one of the things about breath is you are reflecting your environment. You are, the best way of saying it, you're a walking dosimeter. And this slide shows you the process of a single breath. The first part of the breath, this pointer doesn't work. Oh yes. This part is where you're breathing out through the dead space of your lung, through your trachea. And then what is happening here is that I'm plotting carbon dioxide versus exhalation in volumes. And this is when you start to breathe, carbon dioxide increases because you're starting to get dilution of the dead space with air from your deep lung. And then finally you'll reach the plateau, and this is known, the plateau, phase 3 is known as the end stage, end tidal breath. And then phase 4 is inhalation again. What this means is that for a significant breath, the composition of your breath changes throughout your breath. When you're breathing out what you've just breathed in. And then you start to breathe out molecules that are coming from your respiratory tract and then finally from the deep lung. So during the course of a breath cycle, your chemistry changes, not only in composition, but also in the chemistry. And the molecules that come from the end tidal breath, the end of the breath, originate from biochemistry processes that occur within the oral nasal cavities, the pulmonary system, and organs and tissues throughout the body. Final thing is, the composition of the breath will change as a function of the mechanics of how you breathe. Normally, when you ask a person to breathe, when you're breathing normally, you're not aware that you're breathing. You ask a person to breathe for you, and automatically their breathing changes. And as a result of which you hyperventilate. You're inclined to breathe harder. And it's a gender thing. Males tend to breathe much harder than females when you ask them to collect a breath. The data's solid. It's a male thing. It's testosterone related. Okay, so if you want to collect a breath from a sample, a person, the first thing you have to do is be able to collect a breath sample, because if you grab a breath sample from somebody, you end up with garbage, and you do a great analysis of garbage. And so, this is the worst place to start doing science. So what you do is you try and monitor how a person breathes. And the way we did this, and this is the way that we probably set the stage for, is we monitor every tidal breath, how much the volume of the breath a person breathes. We monitor the concentration of carbon dioxide. We monitor the mouth pressure, because if you want to collect a breath sample, you want a person to breathe through a mouthpiece. And the only way you can get a pressure in the mouth is if you make a seal. And finally, we monitor pulse, because that's a good idea of heart rate. And we monitor oxygen sats, which is the amount of oxygen in the blood. And we monitor sample multiple breaths. This is the system that we developed at Hopkins. A subject breathes through the mouth, through a non-rebreathing valve, through a flow meter and a filter, and the breath goes, the inspiratory air comes in here from the far right, comes into the mouth. It's a two-way valve, and it breathes out on the far side. Just to show you how we do this, this is actually me collecting a breath sample. It was a little bit of time ago. I had more hair, and this is on an Air Force base where we were collecting breath from an Air Force worker, and we were using it to assess exposure to jet fuel. But this is what we were monitoring. We use the person acting as his own control. We actually make them follow their breathing in real time. The top curve is their tidal breath. The lower curve is their end tidal CO2. And the second one is the mouth pressure. And when you breathe in, you get a pulse of breath. You get a pulse of pressure. And the bottom is your expiratory end tidal carbon dioxide. Just to belabor the point, this is the data we were collecting with that above equipment. And we asked the person, don't worry about looking at the screen, don't worry about listening to the metronome, which is telling you to breathe in at a defined time. Just breathe normally. This is a person breathing normally. On the top file, what I'm showing is the tidal volume of each of their breaths. And this person weighed about 180 pounds. He's breathing a tidal volume of 1.4 liters, so he's hyperventilating from day one. And what you should notice is the concentration of his carbon dioxide at the end of the breath is much lower than when it started. And what has happened is he's hyperventilated, and he's actually decreased the concentration of CO2 and also he's changed the pH of his blood. Okay, now I'm going to talk a little bit about the chemistry of breath. And what is in breath? And there are three groups of molecules in breath. The first group is molecules or their metabolites originating from inhaled air. And this can be current, what you just breathed in, or historical. Your breath gives a history of what you've done. It can be as long as a month of what you've done. It can be as short as a few hours. But definitely, if you've pumped gas this morning when you came to college, you will be still breathing out gasoline right now. If you took a shower, you will also be breathing out halothane that went through your skin during taking a shower. That's a fact of life. That's everything that's in your breath. And you will breathe out everything you've eaten as a result. So that's a huge thing. And that's the most concentrated source of molecules. And this could be the direct molecule or its metabolite. And then the molecules that are derived from food and beverages. And when you think about breath analysis, everybody thinks about the breath test for drunk driving. But you are, in fact, it's a no-brainer. You know when a person's eating garlic. You know when a person's eating onions. And it's a fact, it's there because of all the sulfur compounds. And finally, the molecules that are produced by anabolic or catabolic reactions throughout the body. That is also in the breath. But this is in the lowest concentration. So what I did is, and there's a quiz at the end to identify the physiological basis of all these molecules. Everyone in this room is breathing out these molecules. So some of them are quite surprising. You're breathing out things like ethylene, carbonyl sulfide, carbon monoxide. This is being produced physiologically. And this is their concentration in volume. So clearly you have products of respiration like carbon dioxide, and you've got products of gut bacteria. This is another list of molecules that we found. Surprise you that people are breathing out hydrogen cyanide, hydrogen sulfide, and this is not people with bad breath because of bad teeth. This is as a result of normal physiology. You breathe out ammonia. So these are molecules everyone in this room is breathing out right now. And these are all produced endogenously as a result of your system. Okay, I'm going to put up a controversial slide because I think very strongly about this. Unique biomarkers in breath can only result from the ingestion, inhalation, or dermal absorption of foreign substances, or from bacteria, viruses, fungi, metabolism. Everything else, what you're looking for is a change in concentration if you're looking at disease. What happens if you have a disease, like if you have diabetes, you increase the concentrations of ketone bodies. Liver disease, you can increase concentrations of sulfur compounds, ammonia. But it's above a normal level of a population. So it's concentration-based differences on the disease diagnosis. Because I have spent 25 years looking at reactive oxygen species, I wanted to just give you a little bit of data. Oxidative stress is involved in every tissue of our body. You, in fact, are generating about 0.3 moles of oxygen, reactive oxygen species per day. Because you use about 14.7 moles of oxygen per day. This is, once again, this is a 75 kilo individual. And reactive oxygen species are involved in signaling, involved in organ dysfunction as a result of disease. They're involved in killing. And in fact, they're probably responsible for normal aging and the fact that people get disease as a result as they get older. What happens is the antioxidant defense mechanism decreases with age. And you can look at oxidative stress, markers of oxidative stress, by looking at markers of damage, which is lipid peroxidation. And you can measure concentrations of molecules such as ethane, ethylene, pentane, aldehydes, etc. in the breath. And we use this for a lot of our research for many years. I just put up a slide here. These increased concentrations of reactive oxygen species are involved in every one of these diseases, whether it's diseases of prematurity or the disease of aging, ALS, Lou Gehrig's disease, scleroderma, infections, you name it, they're involved. And there isn't a single disease that you can think about that does not involve changes in reactive oxygen species. Now, is it the cause or the resultant or the process? It's involved. But I didn't think I would talk about disease because I was coming to a chemistry department. I thought I'd talk about exercise. Everybody thinks about exercise and thinks about disease, reactive oxygen species, oxygen species and exercise. And so what we did was we got a group of people that we asked them to exercise on a stationary bicycle, and we injected technetium into their vein, and we scanned their heart with a gamma camera. So while they were exercising, we were scanning their heart, and we made them, we had a defined protocol, and we wanted to look at exercise and the molecules produced as a result of the exercise. And so we were monitoring the ventilation, the minute ventilation, the oxygen consumption, the CO2 production, the anaerobic threshold. We also measured their cardiac output. And we used what's called a mugger scan, which is a way that you can actually gate the gamma camera to the heart rate, and so that the heart appears to be stationary. So you can look at things like the stroke volume, how much is the heart changing, and you can measure all the properties, so we can get all the information on cardiac output. So this is data from one of the subjects, and what I'm showing here is the blue is the cardiac output, and the red is the minute ventilation, how much they're breathing. And what you should notice here is, as a result of exercise, and the bottom is exercise, and what we ask the person to breathe, to pedal against a given resistance for 3 minutes, and then we increase the pressure against exercise, and then we ask them for another 3 minutes. We did this, and we did this until such time as either their blood pressure went astronomical, or they said, I can't do anymore, or their heart rate was outrageous. And we studied 20 people. This is an incredibly expensive experiment. This costs about $120,000 to do. But this shows you, what you should notice here is that cardiac output changes much more slowly than minute ventilation. So that it starts to uncouple. And if you think about what's in your breath, you're getting, what are your lungs for? It's for the exchange of molecules from your blood to your breath, on the other side of the membrane. So if you change the ratio of cardiac output and minute ventilation, you're actually going to change the way a person's exchange. And so what this is the resultant of 20 people. And what we did was we normalized everybody to his own first sample, and then we looked at their evolution of breath for this molecule, isoprene, which is produced in the cholesterol biosynthetic pathway of cholesterol. And what you should notice is, as a result of exercise, what happens is you blow down your level of cholesterol in your breath. You also blow down the level of isoprene in your breath. You also blow down the level of isoprene in your blood. And what you do then is you change the biosynthesis of cholesterol as a result. But what you see is, and this is blowing it down because you're ventilating the lung much more rapidly than you're having cardiac output. Whereas ethane, it didn't change. It's essentially was constant throughout. So this means that exercise, you don't generate more radicals. All you do is, because the generation of radicals comes through the mitochondria and the conversion of oxygen to water, you don't change that. And so there's no net product generation of reactive oxygen species. The reason is because we've normalized the CO2. But what is interesting is that if you looked at acetone, and what you should notice is this point over here at 124, this is when you've switched to anaerobic. And this tells, once you start to generate acetone, it tells me when you've started to actually work and actually start to burn fat. So this is showing how you're actually, when exercise is going to be useful to burn fat. Most of the time you're not doing anything, just, it's just an exercise. You're not actually losing weight as a result of exercise. This is another study that we did, and this was done with 130 people. By the way, it wasn't done with students. Students would love to do this study. What we did was we fed people for a significant period of time. We actually fed them for 11 weeks. That meant they came to a lab every day and ate a meal, and then took food away for 2 more meals, and they weren't allowed to eat anything but what we supply. And what we did was we started everybody off on we gave the diet was completely blinded. It was generated by the USDA. And the first diet that everybody went into was a B diet, which is a typical American diet, which is high in fat, low in fruit and vegetables. And at the end, everybody ate that diet. And at the end of a 3-week period, we then were blinded, and they got either the same diet or a diet that had 5 times the amount of fruit and vegetables, the same amount of fat, or a diet with 5 times the amount of fruit and vegetables or lower fat. And this is known as the DASH diet, which is a diet to control hypertension. So this is the study design. Everybody came in every day for 3 weeks. We then sampled them after they've been on the diet for 3 weeks. And then they came every day for the next 8 weeks, and we sampled them again. And the idea was to see, could we change oxidative stress as a result of simply changing diet? We knew that you control hypertension by one of the diets, which is the DASH diet. And this is the result. The group, each person is his or her own control, so we're looking about what happened after the first run-in period and the same person at the end of the 8 weeks addition. What you should notice is there's a net change in the breath of the diet peak. They were becoming more oxidatively stressed at the end of as a result of going on the diet for an additional weeks. The groups of people who got the A diet with 5 times the amount of fruit and vegetables, or the DASH diet, or both C diet with 5 times the amount of fruit and vegetables, they became less stressed. And so this is actually the first time you could show in a population that you can control oxidative stress simply by the food they eat. We collect breath from people. We also I've collected breath from horses, dogs, cats. We do routinely from rodents, and collecting breath from rodents doesn't represent anything. This is a totally survivable scenario. You can collect breath, and you only have to train them to do this. And we've studied rats for years without any harm to the animal. We actually collect their breath, and you can train them to do the breath. And this is a study that we did in collaboration with the National Institutes of Aging, where we studied rats that have been they've been given what's called a diet restricted, and they were restricted by 30% of their diet for a lifetime. Or they're fed ad lib. In other words, they've got as much food as they want. And what you should notice is that an ad lib rat, after 28 weeks, weighed about 290 grams. Whereas the same rat that was diet restricted, in other words, and this is not starvation diet, this is a 30% diet restriction, weighed only 168 grams. But what was interesting, if you normalize their ethane, measurement of their breath ethane, to their body, to their carbon dioxide production, you were able to see that the ad lib rat was more oxidatively stressed than the diet restricted rat. And the diet restricted rat was disease-free after 28 months of life. Whereas the ad lib rat was disease that started with cancer. And the only thing we did was control how much food they were eating. This is another study that we did. This time we were looking at OB mice. These are mice that have leptin deficiency, and their C57 black 6 controls. And what you should notice is these are 24-week rats. The obese rat is double the weight of the lean control. And we thought that we would find more oxidative stress in these individuals, but we didn't. What we found was the only molecule that changed was that the obese rat, the obese mouse, was generating ethanol. And we were interested to know why were they generating ethanol, and the obvious thing was gut bacteria. So what we did was we fed the rats, the mice, neomycin, which is an antibiotic which is poorly absorbed, and the only thing it does is it kills gut bacteria. And what happened is the ethanol level dropped. So then what we did was we looked in people. And there's a disease for people who are obese, which is a big problem in America right now. You have increased hypertension, you have increased incidence of diabetes, but you also have increased incidence of fatty liver disease, where the liver makes a lot of fat. And that is typically found in alcoholics. But these people aren't alcoholics, they don't drink. What is happening is their gut is generating ethanol. And that ethanol is causing them to have fatty liver disease. And so what we've done is we've shown that you can control how people get problems with non-alcoholic steatohepatitis as a result of being obese. So if you can change their gut flora by giving them things like yogurt. We published these studies in Hepatology, and this day it was published, my phone rang for the next week from lawyers who thought this was a perfect way of getting people out of drunk driving things because they were obese. Okay, so I picked some examples from my lab. So what I'd like to do now is talk about where we're going in the future. What's the future of it? And breath analysis will have a role in clinical chemistry if it provides new information or or diagnosis, and it provides this information quicker than conventional means. I think anything else, it doesn't propose any advance. So these are the requirements. It provides new information or diagnosis that can be done other ways slower. In other words, when you're sitting in the doctor's office, can you get some results that will tell you, okay, you've got this or that, and therefore I'm going to treat you. So I define real time as getting analysis in 5 to 10 minutes. In other words, while you're still around. And it should be ideally based on a single breath. And there are requirements for this. They must be able to be performed by people with limited training. You can't put a Ph.D. in a clinical lab, and you can't put them in a doctor's office. If you've got, what you're limited to is a clinician who probably has 2 left feet, and no fingers, and is incompetent. And then instruments must be reliable. So that's the requirements. Okay, this is the approach that we use to develop how you collect a single breath. And what this is trying to do is to duplicate a person who is either whistling or playing a musical instrument with a single note. Okay? And so what you do is you ask a person to breathe through a critical orifice that controls the rate at which your breath flows. Because that way you can repeat how a person breathes. And you can't measure flow that well, so you can measure mouth pressure. So because if you have a critical orifice and control, flow is related to pressure. So this is the way we did, and this is the way we designed it. And this is actually an instrument that I designed and is built in Italy. You'll have to look here. The pointer doesn't work. This top one is the CO2 profile of a single breath, and the bottom is the mouth pressure. So you ask a person to breathe at a given mouth pressure, and you actually can get a fixed concentration on a single breath, and you can take a sample and analyze it. I'm going to use an example for some of these studies on looking at breath ammonia. And this is ammonia that you all produce, and you produce it worse if you have liver disease or urea cycle abnormalities, or you have problems with your kidneys. And so ammonia has a huge problem, and it's very difficult to measure blood ammonia. You can measure breath ammonia much easier. This is a busy slide. This is an instrument that we built in collaboration with a group at Rice University, and we measure ammonia in real time using quartz-enhanced photoacoustic spectroscopy, QAPS. And what this is, we measure using a semiconductor laser in the mid-infrared, we measure the photoacoustic effect of ammonia inside a field, and the field is the tuning fork of a watch. And so and the size of the cell is 1 centimeter cubed, and we measure it, the cell pressure is about 100 torr, and we're monitoring we monitor 3 different wavelengths with 3 different lasers. And we're sampling breath at a rate of 100 mils a minute. This is a single breath of ammonia. The first one is the CO2 profile. The airway pressure is the second one on the left, and then the right is the ammonia in a single breath from a person. This person was breathing out around 300 parts per billion of ammonia. And there's 2 different ways of showing the signal. Now the question was, where does that mouth ammonia come from? And I at first thought that the ammonia was coming from mouth bacteria. But it turns out it doesn't. What we did was we asked a person to collect their breath and then perform a mouth rinse. And they mouth rinse either with water, peroxide, hydrogen peroxide, Mylanta, or Coke. And why did we choose those? Water just because it was an easy thing to wash your mouth with. Peroxide because we thought that mouth bacteria were involved. And so if you wash your mouth with hydrogen peroxide, you will in fact kill bacteria. The third one is to use Mylanta because it's basic. And the fourth is Coke because it's acidic. And what was interesting was that if you rinsed your mouth with Mylanta, you actually generated more. So what was happening is the base was in fact, the ammonia was actually in the mucous membrane of the mouth. And so when you washed your mouth with base, you actually got more ammonia out as a result of liberating the ammonia, or ammonium ion that's in the mucous membrane. Whereas the peroxide dropped it, but not too much, but it rose slowly back to baseline. And the way we did this, by the way, we normalized for each test, we normalized to their baseline. So we're looking at only for each person, this is their own, we're normalizing the effect to their original. So where is this ammonia coming from? And this experiment shows it. This is the same experiment, but we're only washing the mouth with ammonia, or we're washing the mouth with water, and then what we ask the person to do is, this is actually 11 people, we ask them between each breath collection, we either ask them to breathe through their nose or through their mouth. So what is happening is what you should notice is initially it dropped as a result of washing the mouth, and then it started to rise again, but it rose much more slowly if you were breathing through your nose because you weren't getting ammonia into the membranes of the mouth. So you can use breath ammonia to look at liver disease and renal disease, but you have to be very careful that the person hasn't drunk anything for the previous hour, hasn't cleaned his teeth with some base, because there's enough base in toothpaste. They haven't drunk orange juice because it'll change. So you have to be careful. But ammonia, you have to know what the history of that person's been doing in order to collect breath ammonia clinically. Another molecule, obviously I did ethane, and this we use ICOS. Once again, in the mid-infrared, we sampled it. This time we're looking at a large multi-pass system where we have an effective path length of 1,500 meters, and this is done by mirrors where the beam shoots from one mirror to the other. And we were able to measure, this is looking at ethane, carbon dioxide, and carbon monoxide, and we're looking at smokers. A smoker will generate oxygen-free radicals because for every puff of cigarette smoke is 10 to the 15 radicals. So you're generating huge volumes of radicals in the lung. And these are two different people at different times. So you can measure ethane in real time. This is a concentration of around 10 parts per billion. And this is looking at breath of a smoker as a function of time after smoking a single cigarette. The final approach that we've been using is a portable GC mass spec. Using thermal desorption GC mass spec. The whole device weighs a little over 1 kilo. And it is capillary GC. It does thermal desorption. And instead of a normal mass spectrometer, we use a differential mobility spectrometer. The whole device samples, breath samples at 10 mils, 100 mils a minute, and we collect a 10-second breath sample, and we perform a complete analysis of breath in 8 minutes. And you measure negative and positive ions. This is the technique of differential mobility spectrometry. What you do is you have a radio frequency field, and then you have plates, and then only certain ions get to the detector. It's essentially like a normal quadrupole mass spectrometer, except it's based on mobility, ion mobility in the electric field. It's very small, very simple, and the actual nuts and bolts of the instrument is only 4 centimeters squared. So this is actually what the data looks like, the real data. And if anybody has done electrophoresis or 2D gels, this essentially looks like a 2D gel. And what the y-axis is chromatographic time. In other words, the chromatography, gas chromatography. The x-axis is the applied potential in the field. This is a person's breath. And what you should notice is this blue thing is water. What I did here was I sampled an individual for an entire week, every hour waking hour. And this individual was me. And I did everything I would normally do in a week. And this is the variation of my breath over a week. And what you should notice is that the standard deviation of my breath is pretty solid. It doesn't vary much over a week's period. The only exception is the ethanol. And I had a beer. And so that screwed up my standard deviation. But it showed that an intervention. It also, ethanol increases if you have a pancake with maple syrup, because you actually generate lots of ethanol just by eating maple syrup because of the sugar from the gut bacteria. Okay, what I hope I've shown is breath analysis is completely non-invasive. Everybody, you have to, I mean, I have an appointment in the School of Public Health, but I also have an appointment in pathology. And I always joke, the only place I couldn't get breath is in the autopsy suite. Breath is totally non-invasive. People can give multiple times without the risk to the subject. There is a limit to how much blood you can get from a subject. There's no limit to the amount of breath. Children give samples willingly. I have sampled children routinely without any problem. And we've collected breath from the neonate. The smallest individual we sampled was a 600-gram, 24-week neonate. And the oldest person was 95 years old. And you could theoretically go from a mouse to an elephant. We haven't gone to an elephant, but we have collected breath from horses. And breath can be collected in the field, in a clinic, inpatient, outpatient, or operating room, or intensive care unit. The advantage is, unlike blood, where you have to refrigerate the sample, breath doesn't need any of that. You can just collect it and do the analysis. Okay, what is the status of breath analysis right now? Breath analysis is used to measure your breathing by carbon dioxide. If you don't use it, it's almost medical malpractice not to use a capnograph. You can measure hydrogen and methane to look at gastrointestinal abnormalities, such as GI transit time, overgrowth, intestinal stasis, or allergies to milk and lactulose. Airways, at reactivity, you can measure nitric oxide. Remember, nitric oxide was the molecule of the year in the 90s, and airways, if you're an asthmatic, you produce much more nitric oxide than you do any other gas. And you can look at metabolism of enzymes in the body. You can titrate any enzyme system. The only requirement is you give the person a labeled derivative that is the substrate for that enzyme, because then you can measure labeled CO2. That's what we do right now. Where are we going? You can measure carbon monoxide to look at neonatal jaundice. You can look at oxidative stress. You can look at cholesterol biosynthesis, because during the biosynthetic pathway you produce isoprene. You can look at renal function with ammonia and alkylamines, liver function. You can look at host response to infection, because as a result of being infected, you increase your oxidative stress. Or you can look at molecules that are produced by the bacteria themselves, or the virus. You can look at urea cycle disorders, hepatic encephalopathy, and exercise physiology by breath ammonia. And you can look at diabetes, fasting, and weight loss using acetone. The only requirement is that you know what is normal. Or each person is his or her own control. Because disease is going to be concentration dependent. So you either need to know what's normal, and that's difficult, because that means you've got to sample 50 to 60,000 people. I've sampled probably close to that, but not every molecule. Well, let's finish by saying, clinical breath analysis, are we there yet? Well, progress is being made, but there's still need for real-time breath analysis. Most of the equipment that's being used has been analytical equipment that you have in any chemical lab. So what you're doing is modifying the analytical method to use that equipment. Currently, and within the last 3 or 4 years, instruments have been built designed for real-time analysis. And proof of principle that instruments have been made, but we're still working on new instruments, smaller instruments. The ideal piece of equipment for breath analysis is small enough that you could give to a patient to take home and monitor themselves using personal monitoring, personal dosimetry. There's a totally, I haven't discussed any of the environmental use of breath analysis, but there's another use of breath analysis that is in terms of homeland security. Breath analysis can tell if a person has been making bombs, has been making drugs. You can change your clothes, you can wash, but you can't change your internal dosimetry in your body. That's a use, and there are people that are studying that. Homeland Security, CIA. My work is, I talked about today, has been supported by NIH, the US Air Force, National Science Foundation, and I've collaborated with lots of people. Italy, groups in Massachusetts, groups in Texas, Bethlehem, Pennsylvania, and a group in Massachusetts. And I'd like to acknowledge my colleagues and students at Hopkins who have helped me work on for more than 30 years in breath analysis. Thank you for your attention. I'll be happy to answer any questions.